So let's take a look at the actual anatomy of the various parts of the alimentary canal. And we'll start with the stomach. So the stomach is going to be an enlargement within the alimentary canal. There are distinct regions within the stomach that have distinct functions. The opening of the stomach is going to come from the esophageal sphincter. The esophageal sphincter will lead to what's referred to as the cardiac region. The cardiac region is going to sit just inferior to the apex of the heart. To the superior medial border, we have the lesser curvature, whereas to the inferior lateral border, we'll have the greater curvature. Coming from the lesser curvature, we'll have the lesser omentum, which will connect with the ligaments around the liver to stabilize the stomach on the medial aspect. Coming away from the greater curvature will be the greater omentum, which will anchor the stomach with the rest of the alimentary canal within the abdominal pelvic cavity. To the superior lateral margin of the cardiac region, we have what's referred to as the fundic region. The fundic region is the area that's going to allow for regurgitation during peristaltic waves within the stomach. That is going to allow for regurgitation of materials within the stomach away from the cardiac region so that when the esophageal sphincter will relax to allow materials to come in from the esophagus, we do not get regurgitation of the acidic materials into the esophagus. Coming away from the fundic and the cardiac region, in between the curvatures, we get the body of the stomach. The body of the stomach is where we're going to see the largest amounts of rugae. The rugae are going to be the ridged structures that take the place of the circular folds. And we'll go a little bit more detail into what that pertains here in a second. As we start to match up the curvatures on the inferior aspect of the stomach, we get to what's referred to as the antrum. The antrum leads to the pyloric canal. The pyloric canal is going to narrow down into the pyloric region. The pyloric region will then narrow down into the two sphincters that make up the pyloric sphincter, which will connect the stomach to the duodenum. So if we look at the rugae within the stomach in a little bit more detail, what we end up getting is this rigid structure of mucosa and submucosa. This rigid structure, unlike the circular folds that will allow for flattening out in the rest of the alimentary canal, we don't want to have this flattening out because coming away from the gastric pits will be a highly acidic hydrochloric acid solution, where if we do not have the proper parts of the mucosal layer, we could actually start to etch away the tissue and get into the submucosal vasculature, leading to issues of sepsis that come into play. That is one of the issues that can come about from excessive ulcerification within the stomach. Notice we have slightly larger mucosa fawns or villi projections coming away from the muscularis mucosa relative to what we saw in the initial picture. We have irregular shapes within these villi formations that form what's referred to as the gastric pit. The gastric pit is going to be made up of several types of cells, which we'll get to here in a second, that are going to be producing all of the gastric juices that are necessary for chemical digestion within the stomach. The principal element within this gastric juice is hydrochloric acid, which is going to take and modify the chemical nature of the material, weakening the chemical attraction and turning solid into liquid. Even though we're turning solid to liquid, we're not technically mechanically breaking down anything. We are chemically breaking down. We do not mechanically break down anything within the stomach. We don't have any structures within the stomach so as to induce mechanical breakdown. Now, if you notice, there is a third layer of muscle within the muscularis layer that we didn't see previously, and that is the oblique layer. And so as we talked about in terms of the peristaltic motion, what this oblique layer does, it allows for a twist to take place within the peristaltic contraction. And what that twist does is it allows for the regurgitation of materials to take place within the stomach itself, leading to the development of the chyme. So if we go a little bit deeper into what we see in the rugae, we end up getting these strong muscular mucosal layers, followed by a very large rigid submucosa. followed by the 3 distinct layers of muscle tissue, leading to the serral layer on the outermost edge. If we were to take that rugal area of mucosa and actually look at what's going on in terms of the gastric pits, we have 5 distinct types of pit cells that are going to come into play. Now, the thing is that we actually break the pit up into 2 parts. We break it up into what's referred to as the neck pit part, and we break it down into what's referred to as the gland pit part. The neck pit part is primarily going to be producing a mucus from the mucous neck cells. This is an acidic mucus, and the mucus that's being produced here is acidic so as to not allow for neutralization to take place prior to etching of the food bolus that's coming into the stomach. Lining the pit are mucus cells that are going to be producing an alkalinic mucus that's there to protect the gastric pit villi from etching and digestion from the the acidic material coming away from the pit. We have 3 types of cells that are going to be producing stuff in terms of one regulating, and that's the G cell producing gastrin, and then 2 cells are going to be producing digestive materials. Those are the parietal cells and the chief cells. The parietal cells are going to be producing what are referred to as intrinsic regulatory factors, as well as the hydrochloric acid. And then the chief cells are going to be producing pepsinogen. Pepsinogen, when it combines with the hydrochloric acid, is going to convert into pepsin. Those 2 materials are necessary for the digestion of proteins within the alimentary canal. So if we go ahead and take a look at these cells in terms of a histological image, this is what we're going to look at. And so notice how we have the lining cells where we have the endoenteric cells, the gastrin producing cells at the very bottom of the pit. They'll be regulating the parietal cells in terms of their production of hydrochloric acid. And deep to the parietal cells will be the chief cells producing the pepsinogen, which will head into the pit itself. Notice how we have the mucus producing cells towards the surface. The ones on the surface mucus cells producing that alkalinic mucin, whereas the neck mucus cell producing the acidic. Notice how the pits are of different depth and different arrangements. And notice the amount of mucosa that surrounds it. That amount of mucosa is going to hopefully stay rigid based off of cellular division and types of connective tissue that we'll see, so as to ensure that we do not have flattening of the rugae, so as to prevent etching of materials within the stomach or ulcerification taking place. The next part of the alimentary canal is the intestines. We break the intestines up into 2 parts, the small intestinal part and the large intestinal part. The small intestinal part is small in diameter. It's very small in diameter, however, it's very long in length. Remember, it's that 5.5 to 6 meters in length. However, it has a very small internal lumen, and that lumen is very filled with villi and microvilli. It's the highest villiated and highest microvilliated region within the alimentary canal, so as to allow for the greatest surface area of interaction to increase the amount of digestion and absorption that will take place. In terms of the gross anatomy of the small intestines, we break it up into 3 parts. The first region is a few centimeters in length, and that is the duodenum. The duodenum will lead to the jejunum. The jejunum is going to be the next 4 to meters of small intestine. The duodenum and the jejunum are going to be the continuation of chemical digestion that began with exposure to foods with saliva and salivary enzymes, followed by the enzymes and the acid within the stomach. As we move through the jejunum, the amount of chemical digestion starts to slow. However, the amount of absorption starts to increase. That amount of absorption is going to continue as we go into the ileum. The ileum is that final third of the small intestine, which is going to lead to the ileocecal sphincter and the beginning of the large intestine. Within the small intestine, we're going to have various structures that are going to be associated with lymphatic responses, because remember, this is inside the body, but not really inside the body. And so we have to somehow protect through immune functions the body from what we're having to digest and absorb. There are a few functions that all three, the MALT, the mucus-associated lymphatic tissue, prior patches, and lacteal will all serve. They will all be involved with mucus production and with immune responses. However, the lacteals have a secondary function as it relates to absorption of materials, and they're going to be involved with the transportation of lipids from the villi, the microvilli of the small intestine, to the liver for processing into circulating lipid tissues. So if we look at what we're talking about here, here is the lacteal. Lacteal is going to be surrounded by the arterioles and capillaries within the villi. This lacteal is going to associate with circulation of lymph from the villi itself into the MALT. That MALT will then lead to its afferent pathway heading out into the liver. Connected with the MALT will be the prior patches. The prior patches are clusters of lymphatic tissue that houses T cells and B cells as well as macrophages within the actual mucosal layer. What these prior patches and lymphatic tissue is going to do is it's going to initiate immune responses to any pathogenic antigen that we might be attempting to digest and absorb during interactions with the food materials. These same tissues can come into play when looking at food allergies and food intolerances, particularly with materials that cannot be readily digested due to irregularity of exposure. The final segment of the intestines or the alimentary canal is the large intestine. So the large intestine is going to be much larger in diameter, but have less villi and is much actually shorter than the small intestine happens to be. So even though we call it large intestine, what we're actually talking about is we're talking about the diameter of the intestine itself and not the length of the intestine. We have less villi and less microvilli because we're not actually doing a lot of digestion and a lot of absorption here. However, we do have a large amount of microbiome that will be alive within the large intestine. Most of this microbiome is going to be seeded from the veniform appendix. The veniform appendix is one of those things that for the longest time we thought was a vestigial organ. The problem is that evolutionary speaking, it's not seen in a lot of other primates. It appears to be more of a symbiotic adaptation to assist with the microbiome that we have within the intestines themselves. The microbiomes are there to help with the digestion. Any chemical digestion, or excuse me, the largest proportion of chemical digestion that takes place within the large intestine is not from actual enzymes that we're producing, but is from the actual enzymes of the microbiome breaking down the food materials. In the study packet, I've given you a list of all the various types of microbiome that is there, as well as some of the more current ideas as to its use within health for the individuals. So if we look at the large intestine, we start with the cecum. The cecum is going to lead to a small little flexure and ascend. As it ascends, it's going to hit the liver and no longer be able to ascend, at which point in time it's going to turn. That turn within the large intestine is referred to as the hepatic flexure. We then get to the transverse colon. The transverse colon will then hit the spleen and costals on the opposite side, at which point in time it's then going to turn again, and this time here we have the splenic or gastric flexure. That is going to descend along the lateral margin of the abdominal cavity, at which point in time it's going to hit the pelvic structures and the pelvic quote-unquote floor and start to curve in the posterior superior angle known as the sigmoid colon. We call it the sigmoid colon because it looks like a little bit of an S. And so most things that look like an S in biology we call sigmoids. The sigmoid colon will hit the posterior edge of the pelvic structures and turn inferiorly, forming the rectal sac. The rectal sac will junction with the anus through the anal sphincter. The last third from the sigmoid colon through the rectum is where fecal matter is formed through compaction due to absorption of most of the water within the materials that happens to be within the rectum and the sigmoid colon. Now, since we lose a lot of the villi and circular folds, we don't have a lot of stuff to hold the large intestine rigid. In this case here, we use two external structures. The two external structures that we're going to be using are related to the tinea coli, and the formation we'll refer to as the haustras. The haustras act like little sacs, and what they do is they segment out the large intestine, whereas pressure within that haustra increases, it propels the materials forward from the cecum towards the rectum and the anus.